kinks and fetishes and behaviors, and most of which probably has never been documented because most of this happens in private. And here I always say on this podcast, anytime we're talking about sexual behavior in humans, we're always making the presumption that it's consensual, age-appropriate, context-appropriate, and species-appropriate. Let's say we're talking about a lot of different species. With that said, just to set context, I was watching this video of ferrets mating, and it's quite violent, actually. There's a lot of neck biting, there's a lot of squealing. If I were going to project and anthropomorphize, I'd say it's not really clear they both want to be there. One would make that assumption. And of course, we don't know. We have no idea. Could be the ritual. It seems to me that there is some crossover of aggression and mating behavior circuitry during the act of mating. And do you think that reflects this sort of stew of competing neurons that are prioritizing in real time? Because of course, as states, they have persistence, as you point out, and you can imagine that states overlapping, 4 different states, the motivational drive to mate, the motivational drive to get away from this experience, the motivational drive to eat at some point, to defecate at some point. All of these things are competing, and what we're really seeing is a bias in probabilities. But when you look at mating behavior of various animals, you see an aggressive component sometimes, but not always. Is it species specific? Is it context specific? And more generally, do you think that there is crosstalk between these different neuronal populations, and the animal itself might be kind of confused about what's going on?
Right. Great questions. I can't really speak to the issue of whether this is species specific because I'm not a naturalist or a zoologist. I've seen, like you have in the wild, for example, lions when they mate. I've seen them in Africa. There's often a biting component of that as well. One of the things that surprised us when we identified neurons in VMH-VL that control aggression in males is that within that population, there is a subset of neurons that is activated by females during male-female mating encounters. Now, you don't generally think of mouse sex as rough sex, but there is a lot of what superficially looks like violent behavior sometimes, especially if the female rejects the male and runs away. And there's some evidence that those female selective neurons in VMH are part of the mating behavior. If you shut them down, the animals don't mate as effectively as they otherwise would. What happens when you stimulate them, we don't yet know because we don't have a way to specifically do that without activating the male aggression neurons. But I think they must be there for a reason because VMH is not traditionally the brain region to which male sexual behavior has been assigned. That's another area called the medial preoptic area. And there we have shown that there are neurons that definitely stimulate mating behavior. In fact, if we activate those mating neurons in a male while it's in the middle of attacking another male, it will stop fighting, start singing to that male, and start to try to mount that male until we shut those neurons off. So those are the make love, not war neurons, and VMH are the make war, not love neurons. And there are dense interconnections between these two nuclei, which are very close to each other in the brain. And we've shown that some of those connections are mutually inhibitory to prevent the animal from attacking a mate that it's supposed to be mating with, or to prevent it from mating with an animal it's supposed to be attacking. But it's also possible that there are some cooperative interactions between those structures, as well as antagonistic interactions. And the balance of whether it's the cooperative or antagonistic interactions that are firing at any given moment in a mating encounter, as you suggest, may determine whether a moment of coital bliss among two lions may suddenly turn into a snap or a growl and a baring of fangs. We don't know that, but certainly the substrate, the wiring is there for that to happen.
I'm sure people's minds are running wild with all this. I'll just use this as an opportunity to raise something I've wondered about for far too long, which is I have a friend who's a psychiatrist who works on the treatment of fetishes. This is not a psychiatrist that I was treated by. I'll just point that out. But they mentioned something very interesting to me long ago, which is that when you look at true fetishes and what meets the criteria for fetish, that there does seem to be some what one would think would be competing circuitry that suddenly becomes aligned. For instance, avoidance of feces, dead bodies, feet, things that are very infectious. Typically, those states of disgust are antagonistic to states of desire, as one would hope is present during sexual behavior. Fetishes often involve exactly those things that are aversive. Feet, dead bodies, disgusting things to most people. And true fetishes in the pathologic sense exist when people have basically a requirement for thinking about or even the presence of those ordinarily disgusting things in order to become sexually aroused. As if the circuitry has crossed over. And the statement that rung in my mind was people don't develop fetishes to mailboxes or to the color red or to random objects and things. They develop fetishes to things that are highly infectious and counter-reproductive appetitive states. So I find that interesting. I don't know if you have any reflections on that as to why that might be. I'm tempted to ask whether or not you've ever observed fetish-like behavior in mice, but I find it fascinating that you have this area of the brain that's so highly conserved, the hypothalamus, in which you have these dense populations intermixed, and that the addition of a forebrain, especially in humans, that can think and make decisions could in some ways facilitate the expression of these primitive behaviors, but could also complicate the expression of primitive behaviors.
Right. I would agree. I think one way of looking at fetishes from a neurobiological standpoint is that they represent a kind of appetitive conditioning where something that is natively aversive or disgusting, by being repeatedly paired with a rewarding experience, changes its valence, its sign, so that now it somehow produces the anticipation of reward the next time a person sees it. Now, I don't know that literature in animals, so I don't know if you could condition a mouse to eat feces, for example, although there are animals that are naturally coprophagic. That is, and maybe mice do that occasionally, I'm not sure. But that is one way to think about it, and that could certainly involve in humans the more recently evolved parts of the brain, the cortex that is sort of orchestrating both what behaviors are happening and whether reward states are turning on in association with those behaviors that are happening. And that's the part that I think is difficult and challenging to study in a mouse, but certainly bears thinking about, because it's a really interesting, again, sort of counterintuitive aspect. Again, like rough sex, people that want to have fighting or violence or aggressiveness in order to be sexually aroused, and fetishes. And in fact, when we made that discovery initially, it raised the question in my mind whether some people that are serial rapists, for example, and engage in sexual violence might in some level have their wires crossed in some way, that these states that are supposed to be pretty much separated and mutually antagonistic are not, and are actually more rewarding and reinforcing. I think it's going to be a long time before we have figured it out. But when you think about it, there is no treatment that we have for a violent sexual offender that eliminates the violence, but not the sexual desire and sexual urge. Whether it's physical castration or chemical castration, it eliminates both.
Definitely an area that I think, human neuroscience in general needs a lot of tools, right, in terms of how to probe and manipulate neural circuitry. I'd love to turn to this area that you mentioned, the medial preoptic area. I'm fascinated by it because just as within the VMH, you have these neurons for mating and fighting, or aggression. My understanding is medial preoptic area contains neurons for mating, but also for temperature regulation. And perhaps I'm making too much of a leap here, but I've always wondered about this phrase "in heat." As certainly the menstrual and or estrous cycle in females is related to changes in body temperature. In fact, measuring body temperature is one way that women can fairly reliably predict ovulation, et cetera. Although this is not a show about contraception. Please rely on multiple methods as necessary. Don't use this discussion as your guide for contraception based on temperature. But if you stimulate certain neurons in the medial preoptic area, you can trigger dramatic changes in body temperature and or mating behavior. What's the relationship, if any, between temperature and mating, or do we simply not know?
I don't know what the relationship is between temperature and mating neurons in the preoptic area. I suspect that they are different populations of neurons because it's become pretty clear that the preoptic area has many different subsets of neurons that are specifically active during different behaviors, even different phases of mating behavior. So there are mounting neurons, there are intromission, thrusting neurons, and ejaculation neurons, and sniffing neurons.
Wait. So I think I've heard this before, but I just want to make sure that people get this. And I want to make sure I get this. So you're telling me within medial preoptic area, there are specific neurons that if you stimulate them will make males thrust as if they're mating.
No. So this is not based on stimulation experiments. It's based on imaging experiments right now that we see when we look in the preoptic area at what neurons are active during different phases of aggression. We see that there are different neurons that are active during sniffing, mounting, thrusting, and ejaculation. And they become repeatedly activated each time the animal goes through that cycle.
During mating.
During the mating cycle. There are also some neurons there that are active during aggression, which are distinct, and we don't know whether those neurons are there to promote aggression or to inhibit mating when animals are fighting. We have some evidence that suggests it may be the latter, but we don't know for sure yet. The thermosensitive neurons are really interesting because you mentioned the phrase "in heat," and then in the context of aggression, you talk about hot-blooded people or hotheads. There's just recently a paper showing there are thermoregulatory neurons in VMH as well. So all of these homeostatic systems for metabolic control and temperature control are intermingled in these nuclei, these zones that control these basic survival behaviors like mating and aggression and predator defense. And I would imagine that the thermoregulation is tightly connected to energy expenditure, and that, again, these neurons are mixed together to facilitate integration of all these signals by the brain in some way that we don't understand to maintain the proper balance between energy conservation and energy consumption during this particular behavior or that behavior. I mean, I've always been fascinated by the question, why is it that violence goes up in the summertime when the temperatures are high? Does it really have something to do with the idea that increased temperature increases violence? It seems hard to believe because we're homeothermic and we pretty much stay around 98.6 Fahrenheit. It could be other social reasons why that happens. People are outside, out on the street, bumping into each other. But I think there could well be something that ties thermoregulation to aggressiveness as well as to mating behavior.
Fascinating. Yeah. I asked in the hopes that maybe in the years to come, your lab will parse some of the temperature relationships. And I realize it could be also regulated by hormones in general. So it's tapping into two systems for completely different reasons. But anyway, an area that intrigues me because of this notion of hot-headedness or cool, calm, and collected. And also the fact that, I probably should have asked about this earlier, that arousal itself is tethered to the whole mating and reproductive process. I mean, without a sort of seesawing back between the sympathetic and parasympathetic arousal, relaxed states, there is no mating that will take place. So it's fascinating the way these different competing forces and seesaws operate. Several times during the discussion so far, we've hit on this idea that the same behavior can reflect different states, and different states can converge on multiple behaviors as well. You had a paper not long ago about mounting behavior, which I found fascinating. Maybe you could tell us about that result, because to me it really speaks to the fact that mounting behavior can, in one context, be sexual, and in another context actually be related to, we presume, dominance. And I think that my friends who practice jujitsu will say, when I talk about that result, they say, of course, mounting the other person and dominating them, there's nothing sexual about it. It's about overtaking them physically, literally being on their neck side as opposed to on their own, lying on their own back. Just fascinating, very primitive, and yet I think speaks to this idea that mounting behavior might be one of the most fundamental ways in which animals and perhaps even humans express dominance and or sexual interactions.
Yep. That's a fascinating question, and it was harder to figure out than you might have thought. So there's been this debate for a long time in the field. When you see two male mice mounting each other, is this homosexual behavior? Is this a case of mistaken sexual identification, or is this dominance behavior? And if you train an AI algorithm to try to distinguish male-male mounting from male-female mounting, it does not do a very good job, because motorically, those behaviors look so similar. And so how did we wind up figuring out that most male-male mounting is dominance mounting? There are two important clues. One is the context. And so male-male mounting tends to be more prominent among mice when they haven't had a lot of fighting experience. And then as they become more experienced in fighting, they will show relatively less mounting towards the other male and more attack. And they'll transition quickly from mounting to attack. And so the mounting is always seen in this context of an overall aggressive interaction. And then the second thing, which, believe it or not, was suggested by a computational theoretical person in my lab, Ann Kennedy, who now has her own lab at Northwestern. She said, well, males are known to sing when they mount females, ultrasonic vocalizations. Why don't you see what kinds of songs they're singing when they're mounting males? Maybe it's a different kind of song. Well, what we found out is they don't sing at all when they're mounting a male. So you can easily distinguish whether mounting behavior by a male mouse is reproductive or agonistic, aggressive, according to whether it's accompanied by ultrasonic vocalizations or not. And it turns out that different brain regions are maximally active during these different types of mounting. So VMH, the aggression locus, is actually active during dominance mounting, and you can stimulate dominance mounting if you weakly activate VMH, whereas MPOA is most strongly activated during sexual mounting, and that's always accompanied by the ultrasonic vocalization. So this shows how difficult and dangerous it can be to try to infer an animal's state or intent or emotion from the behavior that it's exhibiting, because the same behavior can mean very different things depending on the context or the interaction with the animal.
And I would say even more so with when that animal is a human or is multiple humans.
That's right. And there are many examples. Animals show chasing to obtain food, a prey animal that they're going to kill and eat, and they show chasing to obtain a mate that they're going to have sex with. And so the intent of the chasing is completely different, and we don't know in all these cases whether there are separate circuits or common circuits that are being activated.
I'm obsessed with dogs and dog breeds and et cetera, et cetera. And one thing I can tell you is that female dogs will mount and thrust, we had a female pit bull mix, a very sweet dog. But in observing her, it convinced me that one can never assume that male dogs are more aggressive than female dogs. It turns out, in talking to people who are quite skilled at dog genetics and dog breeding, that there's a dominance hierarchy within a litter, and it crosses over male-female delineations. So you can get a female in the litter that's very dominant and a male that's very subordinate, and no one really knows what relates to. This is also why little dogs sometimes will get right up in the face of a big Doberman Pinscher and just start barking, which is an idiotic thing for it to do, but they can be dominant over a much larger dog. Very strange, to me anyway. Female-female mounting. Do you observe it in mice? Are there known circuits? And what evokes female-female mounting or female-to-male mounting if it occurs?
Good. Yes, there are clear examples of females displaying male-type mounting behavior towards other females. We see this most commonly in the lab where we are housing females with their sisters, say 3 or 4 in a cage. We take one out and we have her mate with a male where the male's doing the mounting. Now we take that female and we put her back in the cage with her litter mates and she starts mounting them. Now, what the function of that is, if it has any function, or what it means, what's driving it, we don't know. But we do know that if we stimulate the neurons that control mounting in males in the medial preoptic area, if we stimulate that same population in females, it evokes male-type mounting towards either a male or a female target. In fact, we have a movie where we have a female that has just been mounted by a male. So the male's on top and she's underneath. And we stimulate that region of MPOA in the female, and she crawls out from underneath the male who has just mounted her, circles around behind him, and climbs up on top of him and starts to try to mount him and thrust at him.
That has a name online. It's called a switch.
Is that right?
Don't ask me how I know that.
Okay.
But it's a pretty, it's a term that you hear. You also hear the term topping from the bottom, which it sounds like that is a literal topping from the bottom.
See.
That's more of a psychological phrase from what I hear. I have friends that are educating me in this language, mostly because I find this kind of neurobiological discussion fascinating. And at some point, right, I attempt in my mind to superimpose observations from the online communities that I'm told about and asked about to this. But I should point out, it's always dangerous and in fact, inappropriate to make a one-to-one link. Humans they maintain all the same neural circuitry and pathways that we're talking about today in mice, but that forebrain does allow for context, et cetera.
Yep. So what the function is of female mounting, I don't know. It could be a type of dominance display. It's hard to measure that because people haven't worked on female dominance hierarchies to the same extent that they've worked on male dominance hierarchies, but it indicates that the circuits for male-type mounting are there in females, as early work from Catherine Dulac suggested some years ago.
Fascinating. I love that paper because, as you pointed out for chase, for mounting behavior, we see it and we think one thing specifically. After hearing this result, actually, I'm not a big fan of fight sports. I watch them occasionally because friends are into them, but I've seen boxing matches, MMA matches where at the end of a round, if someone felt that they dominated, they will do the unsportsmanlike thing of thrusting on the back of the other person before they get off, almost like I dominated you, and so mimicking sexual-like behavior, but there's no reason to think that it's sexual, but they're sending a message of dominance is what it implies. I'd love to talk about something slightly off from this circuitry, but I think that's related to the circuitry, at least in some way, which is this structure that I've always been fascinated by, and I can't figure out what the hell it's for, because it seems to be involved in everything, which is the PAG, the periaqueductal gray, which is a little bit further back in the brain for people that don't know. It's been studied in the context of pain. It's been studied in the context of the so-called lordosis response, the receptivity or arching of the back of the female to receive intromission and mating from the male. How should we think about PAG? Clearly, it can't be involved in everything. I'm guessing it's at least as complex as some of these other regions that we've been talking about, different types of neurons controlling different things. But how does PAG play into this? In particular, I want to know, is there some mechanism of pain modulation and control during fighting and or mating? And the reason I ask is that while I'm not a combat sports person, years ago I did a little bit of martial arts, and it always was impressive to me how little it hurt to get punched during a fight and how much it hurt afterwards, right? So there clearly is some endogenous pain control that then wears off and then you feel beat up.
Yep.
Or at least in my case, I felt beat up. What's PAG doing vis-a-vis pain and what's pain doing vis-a-vis these other behaviors?
Good. So I think of PAG like a old-fashioned telephone switchboard where there are calls coming in and then the cables have to be punched into the right hole to get the information to be routed to the right recipient on the other end of it. Because pretty much every type of innate behavior you can think of has had the PAG implicated in it. There's a whole literature showing the involvement of the PAG in fear, different regions of the PAG. The dorsal PAG is involved in panic-like behavior, running away. The ventral PAG is involved in freezing behavior. Both the MPOA and VMH send projections to the PAG to different regions of the PAG. So in cross-section, I hate to say this, but in cross-section, the PAG kind of looks like the water in a toilet when you're standing over an open toilet bowl. Mm-hmm. And if you imagine a clock face projected onto that, it's like the PAG has sectors from 1 to 12, maybe even more of them. And in each of those sectors, you find different neurons from the hypothalamus are projecting. So it could turn out that there is a topographic arrangement along the dorsal ventral axis of the PAG and the medial lateral axis of the PAG that determines the type of behavior that will be emitted when neurons in that region are stimulated. And I think sort of all of the evidence is pointing in that direction, but by no means has it been mapped out. Now, the thing that you mentioned about it not hurting when you got beat up during martial arts, there is a well-known phenomenon called fear-induced analgesia, where when an animal is in a high state of fear, like if it's trying to defend itself, there is a suppression of pain responses. And I'm not sure completely about the mechanisms and how well that's understood, but for example, the adrenal gland has a peptide in it that is released from the adrenal medulla, which controls the fight or flight responses, and that peptide has analgesic activities.
Now, whether what that peptide
is called bovine adrenal medullary peptide of 22 amino acid residues, and I only know about it because it activates a receptor that we discovered many years ago that's involved in pain, and we thought it promoted pain, but it turns out that this actually inhibits pain. It's like an endogenous analgesic. Whether this is happening, this type of analgesia is happening when an animal is engaged in offensive aggression or in mating behavior, I don't know, but it certainly is possible. And I don't know whether these analgesic mechanisms are happening in the PAG. They could also be happening a little further down in the spinal cord. The PAG is really continuous with the spinal cord. If you just follow it down towards the tail of an animal, you will wind up in the spinal cord. And so it could be that there are influences acting at many levels on pain in the PAG and in the spinal cord as well. And it may well be known, I just don't know it. I want to distinguish clearly between things that are not known, that I know are unknown, which is in a fairly small area where I have expertise, from things that may be known, but I'm ignorant of them because I just don't have a broad enough knowledge base to know that.
Sure. We appreciate those delineations. Thank you. PAG is, I think this description of it is an old-fashioned telephone switchboard, and now every time I look into the toilet, I'll think about the periaqueductal gray, and every time I see an image of periaqueductal gray, I think about a toilet. That is an excellent description, because in fact, I drew a circle with a little thing at the bottom, and well, I'll put a post or link to a picture of PAG, and you'll understand why David and I are chuckling here, because indeed, it looks like a toilet when staring into a toilet. Tell us about tachykinin. I've talked about this a couple times on different podcast episodes because of its relationship to social isolation. And in part because the podcast was launched during a time when there was more social isolation. My understanding is that tachykinin, and you'll tell us what it is in a moment, is present in flies and mice and in humans and may do similar things in those species.
That's right. So tachykinin refers to a family of related neuropeptides. So these are brain chemicals. They're different from dopamine and serotonin in that they're not small organic molecules. They're actually short pieces of protein that are directly encoded by genes that are active in specific neurons and not in others. And when those neurons are active, those neuropeptides are released together with classical transmitters like glutamate, whatever. Tachykinins have been famously implicated in pain, particularly tachykinin 1, which is called substance P, one of the original pain modulating. This is something that promotes inflammatory pain. But there are other tachykinin genes. In mice, there are 2. In humans, I think there are 3. And in Drosophila, there is 1. And the way we got into tachykinins is from studying aggression in flies. We thought, since neuropeptides have this remarkable parallel evolutionary conservation of structure and function, like neuropeptide Y controls feeding in worms, in flies, and mice, and in people. Oxytocin-like peptides control reproduction in worms and mice and in people. We thought we might find peptides that control aggression in flies and in people. And so we did a unbiased screen of peptides and found indeed that one of the tachykinins, Drosophila tachykinin, those neurons, when you activate them, strongly promote aggression, and it depends on the release of tachykinin. Now, the interesting thing is that in flies, just like in people and practically any other social animal that shows aggression, social isolation increases aggressiveness. So putting a violent prisoner in solitary confinement is absolutely the worst, most counterproductive thing you could do to them. And indeed, we found in flies that social isolation increases the level of tachykinin in the brain. And if we shut that gene down, it prevents the isolation from increasing aggression. So since my lab also works on mice, it was natural to see whether tachykinins might be upregulated in social isolation and whether they play a role in aggression. And this is work done by a former postdoc, Moriel Zelikovsky, now at University of Salt Lake City in Utah. And she found remarkably that when mice are socially isolated for 2 weeks, there is this massive upregulation of tachykinin 2 in their brain. In fact, if you tag the peptide with a green fluorescent protein from a jellyfish genetically, the brain looks green when the mice are socially isolated because there's so much of this stuff released. And she went on to show that that increase in tachykinin is responsible for the effect of social isolation to increase aggressiveness and to increase fear and to increase anxiety. And in fact, there are drugs that block the receptor for tachykinin, which were tested in humans and abandoned because they had no efficacy in the tests that they were analyzed for. If you give those drugs to a socially isolated mouse, it blocks all of the effects of social isolation. It blocks the aggression, it blocks the increased fear and the increased anxiety. And that Moriel described it, the mice just looked chill. It's not a sedative, which is really important. It's not that the mice are going to sleep. Most remarkably is once you socially isolate a mouse and it becomes aggressive, you can never put it back in its cage with its brothers from its litter because it will kill them all overnight. But if you give it this drug, which is called osanatant, that blocks tachykinin 2, that mouse can be returned to the cage with its brothers and will not attack them and seems to be happy about that for the rest of the time. So this is an incredibly powerful effect of this drug. And I've been really interested in trying to get pharmaceutical companies to test this drug, which has a really good safety profile in humans, in testing it in people who are subjected to social isolation stress or bereavement stress. And this is one of the areas where I learned an eye-opening lesson as a basic scientist who naively thought that if you make a discovery and it has translational applications to humans, that pharmaceutical companies are going to be falling all over themselves to try it. And they're not interested because once burned, twice shy. These drugs were tested for efficacy in schizophrenia. I have no idea why. There's very little preclinical data to suggest that. Not surprisingly, they failed. When a drug fails in clinical trials in phase 3, it costs $100 million to the company that carried out that clinical trial. So there's a huge slag heap of discarded pharmaceuticals, many of them inhibitors of neuropeptide action, that could be useful in other indications, such as the one we discovered, but there's a huge economic disincentive for pharmaceutical companies to test them again, because the conclusion that they drew from all these failed tests, particularly in the 2010s and before that, is that the reason they failed is because animal experiments with drugs don't predict how humans will respond to the drugs. And therefore, we shouldn't try to extrapolate from any other data that we get from animal experiments, mouse or rat experiments, to humans, because they'll lead us down the wrong track. And I think that that is probably wrong. In some cases, it may be right, but in other cases, there's good reason to think, because these brain regions and molecules are so evolutionarily conserved that they ought to be playing a similar role in humans. In fact, there is a paper showing that in humans that have borderline personality disorder, there's a strong correlation between their self-reported level of aggressiveness and serum levels of a tachykinin, in this case tachykinin 1, as detected by radioimmunoassay. This is work of Emil Coccaro, who's a clinical psychiatrist at the University of Chicago. So there is a smoking gun in the case of humans as well. And I was actually trying to interest a pharmaceutical company that was testing these drugs actually for treatment of hot flashes in females, in humans, where there is actually good animal data to think that it might be useful. But I realized that this clinical trial was going on during the COVID pandemic. And I approached them and said, look, nature may have actually done for you the experiment that I want you to do, because some of the people who are getting drug or placebo are going to have been socially isolated, and some of them will have not. Why don't you get them to fill out questionnaires and see whether the ones who were given the drug and socially isolated felt less stressed and less anxious than the ones who were not socially isolated, and they would not touch it, because they're in the middle of a clinical trial for a different indication for this drug, and they have to report any observation that they make about that drug in their patient population. So if they were to ask these questions and get an unfavorable answer, oh my God, I felt even worse when I took this drug and I was isolated, they would be obliged to report that to the FDA, and that could torpedo the chances for the drug being approved in the thing that it was in clinical trials for. So it's better not to ask and not to know. than it is to try to find out more information that could lead to another clinical indication. So I remain convinced that this family of drugs could have very powerful uses in treating some forms of stress-induced anxiety or aggressiveness in humans, but it's just very difficult for economic reasons to find a way to get somebody to test that.
Yeah, a true shame that these companies won't do this, and especially given the fact that many of these drugs exist and their safety profiles are established, because that's always a serious consideration when embarking on a clinical trial. Perhaps in hearing this discussion, someone out there will understand the key importance of this and will reach out to us. We'll provide ways to do that to get such a study going in humans. Because I think if enough laboratories ran small-scale clinical trials, pharma certainly would perk up their ears, right? I mean, they're so strategic, sometimes to their own.
I mean, I would like to say also, I'd like to see this tested on pets. I mean, there's a huge number of pets right now that are suffering separation anxiety because humans bought them to keep them company during the COVID pandemic.
And now they're home alone.
Now they're home alone,
Yeah.
okay? And if this thing works in mice, there's certainly a higher chance it's going to work in dogs or in cats than it is going to work in humans. And if it did, that would be even more encouragement to continue along those lines. People sometimes forget that although we work on animals and we ultimately want to understand humans, we care about how our results apply to the welfare of animals as well, and particularly domestic pets, which is a multi-billion-dollar industry in this country. So if there's ways that they can be made to feel better when they're separated from their owners, that would certainly be a good thing.
Absolutely. We will put out the call. We are putting out the call. And I know for sure there will be a response. Just underscoring what we've been talking about even more, every time we hear about a school shooting, like in Texas recently, or I happen to be in New York during the time when there was a subway shooting, for whatever reason, I listened to the book about Columbine that went into a very detailed way about the origin of those boys and that committed that. And every single time, there's the person who commits those acts is socially isolated. As far as I know, there might be some exceptions there. And sometimes this crosses over with other mental health issues, but sometimes no apparent mental health issues. So social isolation clearly drives powerful neurochemical and neurobiological changes. I really hope that tachykinin 1 and 2, those are the main ones in humans, will be explored in more detail. Also, I didn't know that tachykinin 1 is substance P, and substance P is tachykinin 1.
Yes, tachykinin 1 is the gene name, and tachykinin 2 in humans is called neurokinin B. That's the name of the protein. I just refer to it by the gene name because it makes it easier, and I don't have to keep remembering two names for each thing.
And if I'm not mistaken, you put yourself in the company of geneticists because of your original training was in genetics, immunology, and areas related to that.
It was in cell biology, and I didn't actually have formal training in genetics as a graduate student, but I think I'm a geneticist at heart. That's just the way I like to think about things. And when I started working on flies, I came out of the closet as a geneticist, as it were.
Wonderful. As long as we're talking about humans, I'd love to get your thoughts about human studies of emotion. I know you wrote this book with Ralph Adolphs. You have this new book, which we'll provide a link to, which I've read front to back twice. It's phenomenal. I've mentioned it before on the podcast. It's really, there are books that are worth reading, and then there are books that are important. And I think this book is truly important for the general population to read and understand. And neuroscientists should read and understand the contents, because we, as a culture, are way off in terms of how we think about emotions and states and behaviors. So we'll put a link to that. It's really worth the time and energy to read it. And it's written beautifully, I should say. Very accessible even for non-scientists. There's a heat map diagram in that book that I think about. This is a heat map diagram of subjective reports that people gave of where they experience an emotion or a feeling, a somatic feeling in their body or in their head, or both, when they are angry, sad, calm, lonely, et cetera, et cetera. And I wouldn't want people to think that those heat maps were generated by any physiological measurement because they were not. And yet, I don't think we can have a discussion about emotions and states and the sorts of behaviors that we're talking about today without thinking about the body also.
Yep.
And I'm not coming to this as a Northern California mind body. I've been to Esalen once. I didn't go in the baths. I went there, I gave a talk, and I left. It is very beautiful. If anyone wants to know what it looks like, I think that final scene of Mad Men is shot at Esalen. It's a very beautiful place. And yet, mind-body is to me is a neurobiological construct because the nervous system extends through out of the cranial vault and into the spinal cord and body and back and forth. Okay. How should we think about the body and in terms of states? And at some point, I'd love for you to comment on that heat map experiment because it does seem that there's some regularity as to where people experience emotions. When people are in a rage, for instance, they seem to feel it both in their gut and in their head, it seems, on average. And people love to extrapolate to gut intuition or that the chakras or anger is in the stomach, and this goes to Eastern medicine, et cetera. How should we think about mind, body in the context of states and think about it as scientists, maybe even as neuroscientists or geneticists?
Good. So for the answer to the first question about the heat maps and people associating certain parts of their body with certain emotional feelings, this goes back to something called the somatic marker hypothesis that was proposed by Antonio Damasio, who is a neurologist at USC. The idea that our subjective feeling of a particular emotion is in part associated with a sensation of something happening in a particular part of our body, the gut, the heart. I don't see the liver invoked very much in emotional characterization.
But gall and the gallbladder,
yes,
somebody having a lot of
that's
gall.
right.
I don't know why I make a fist when I say that, but I'm guessing the gallbladder is shaped like a fist.
That's right. And if there is a physiology underlying these heat maps, it could reflect increased blood flow to these different structures. And that in turn reflects what you were talking about, that is, emotion definitely involves communication between the brain and the body, and it's bidirectional communication, and it's mediated by the peripheral nervous system, the sympathetic and the parasympathetic nervous system, which control heart rate, for example, blood vessel, blood pressure, and those neurons receive input from the hypothalamus and other brain regions, central brain regions, that control their activity. And when the brain is put in a particular state, it activates sympathetic and parasympathetic neurons, which have effects on the heart and on blood pressure. And these in turn feed back onto the brain through the sensory system. And a large part of this bidirectional communication is also mediated through the vagus nerve, which many of your listeners and viewers may have heard about because it's become a topic of intense activity now. People have known for a long time. So the vagus nerve is a bundle of nerve fibers that comes out basically of your skull, out of the central nervous system, and then sends fibers in to your heart, your gut, all sorts of visceral organs. So when you have and that information is both, you used the words earlier in our discussion, afferent and efferent. So the vagal fibers sense things that are happening in the body. So when the reason you feel your stomach tied up in knots if you're tense is that those vagal fibers are sensing the contraction of the gut muscles. And they're also afferents, which means that information coming out of the brain can influence those peripheral organs as well. And there's work from a number of labs just in the last 6 months or so where people are starting to decode the components of the different fibers in the vagus nerve. And it's amazing how much specificity is. There are specific vagal nerves that go to the lung, that control breathing responses, that go to the gut, that go to other organs. It's almost like a set of color-coded lines, labeled lines for those things. And now how those vagal afferents play a role in the playing out of emotion states is a fascinating question that people are just beginning to scrape the surface of. But I think what's exciting now is that people are going to be developing tools that will allow us to turn on or turn off specific subsets of fibers within the vagus nerve and ask how that affects particular emotional behaviors. So you're absolutely right. This brain-body connection is critical, not just for the gut, but for the heart, for the lungs, for all kinds of other parts of your body. And Darwin recognized that as well. And I think it's a central feature of emotion state, and I think what underlies our subjective feelings of an emotion.
Incredible. David, I have to say, as a true fan of the work that your lab has been doing over so many decades, and first of all, I was delighted when you stopped working on stem cells, not because you weren't doing incredible work there, but because I saw a talk where you showed a movie of an octopus spitting out, or not spitting, but squirting out a bunch of ink and escaping, and you said you were going to work on things of the sort that we're talking about today: fear, aggression, mating behaviors, social behaviors. It's been incredible to see the work that your lab has done, and I know I speak on behalf of a tremendous number of people when I say thank you for taking time out of your important schedule to share with us what you've learned. My last question is a simple one, which is, will you come back and talk to us again in the future about the additional work that's sure to come?
I would be happy to do that, and I really have appreciated your questions. They've all been right on the money. You've hit all of the critical, important issues in this field, and you've uncovered what is known, the little bit is known, and how much is not known. And I think it's important to emphasize the unknown things, because that's what the next generation of neuroscientists has to solve. And so I hope this will help to attract young people into this field, because it's so important, particularly for our understanding of mental illness and mental health and psychiatry. We've got to figure out how emotion systems are controlled in a causal way if we ever want to improve on the psychiatric treatments that we have now. And that's going to require the next generation of people coming into the field.
Absolutely. I second that. Well, thank you. It's been a delight.
Thank you. Great. Really appreciate it.
Thank you for joining me today for my discussion with Dr. David Anderson. Please also be sure to check out his new book, The Nature of the Beast: How Emotions Guide Us. It's a truly masterful exploration of the biology and psychology behind what we call emotions and states of mind and body. If you're learning from and or enjoying this podcast, please subscribe to our YouTube channel. That's a simple, zero-cost way to support us. Please also subscribe to the podcast on Spotify and Apple. And on both Spotify and Apple, you have the opportunity to leave us up to a 5-star review. If you have questions or comments or suggestions about topics you'd like us to cover or guests you'd like us to interview on the Huberman Lab podcast, please put those in the comment section on YouTube. We do read all those comments and we do take them to heart. Please also check out the sponsors mentioned at the beginning of today's podcast. And check out Momentous Supplements, our new partners in the supplement space, and check out Athletic Greens. That's the best way to support this podcast. If you're not already following us on social media, please do so. We are Huberman Lab on Twitter, and we are also Huberman Lab on Instagram. And both places, I cover science and science-related tools, some of which overlap with the content of the Huberman Lab podcast, but much of which is unique from the content covered on the Huberman Lab podcast. Again, that's Huberman Lab on Instagram and Huberman Lab on Twitter. Please also check out our Neural Network monthly newsletter. This is a newsletter that has summaries of podcast episodes. It also includes a lot of actionable protocols. It's very easy to sign up for the newsletter. You go to hubermanlab.com, click on the menu, go to newsletter. You supply your email, but we do not share your email with anybody. We have a very clear and rigorous privacy policy, which is we do not share your email with anybody. And the newsletter comes out once a month, and it is completely zero cost. Again, just go to hubermanlab.com and go to the Neural Network newsletter. I'd also like to point out that the Huberman Lab podcast has a clips channel. So these are brief clips, anywhere from 3 to 10 minutes, that encompass single concepts and actionable protocols related to sleep, to focus, interviews with various guests. We talk about things like caffeine, when to drink caffeine relative to sleep, alcohol, when and how and if anyone should ingest it relative to sleep, dopamine, serotonin, mental health, physical health, and on, all the things that relate to the topics most of interest to you. You can find that easily by going to YouTube, look for Huberman Lab clips in the search area, and it will take you there. Subscribe, and we are constantly updating those with new clips. This is especially useful, I believe, for people that have missed some of the earlier episodes, or you're still working through the back catalog of Huberman Lab podcasts, which admittedly can be rather long. And last, but certainly not least, thank you for your interest in science. Not necessarily, I can do it by just kind of moving my jaw now because I've done a little bit of diving. But what's the story there? We don't have to get into all the differences in atmospheric pressure, et cetera. But if I'm taking off and my ears are plugged, or I've recently ascended, plane took off, my ears are plugged, do I plug my nose and blow out, or do I plug my nose and suck in?
Right. So the basic idea is that if your ears feel bad because you're going into an area of higher pressure, so if they pressurize the cabin more than the pressure that you have on the surface of the planet, your eardrums will be bending in, and they don't like that. If you push them more, they'll hurt even more.
That's a good description, that that the pressure goes up, then they're going to bend in.
Bend in. And then the reverse would be true if you go into an area of low pressure. So if you're starting to drive up the mountainside, the pressure is getting lower and lower outside. Now the air behind your eardrum is ballooning out, right? So it's just a question of, are you trying to get more pressure or less pressure behind the eardrum? And there's a little tube that does that and comes down into your, back of your throat there. And if you force pressure up that tube, you're going to be putting more air pressure into the compartment
to counter it.
If it's not enough, and if you're sucking, you're going the other way. In reality, I think as long as you open the passageway, I think the pressure differential is going to solve your problem. So I think you could actually blow in when you're not, quote, supposed to.
Okay, so you could just hold your nose and blow air out, or hold your nose and suck in the effect. Either way is fine.
I think so.
Excellent. I just won $100 from Harvey Carton. Thank you very much. This is a lot. Harvey and I used to teach neuroanatomy together. And I was like, I don't think it matters, but thank you, Bruce. I'll split that with you.
Okay.
This is important stuff. But it's true. You hear this, so it doesn't matter either way.
I'm no expert in this area.
Don't worry.
Don't quote me.
He's not going to. Well, I'm going to quote you. But okay, so we've talked about the inner ear and we've talked about the cerebellum. I want to talk about an area of the brain that is rarely discussed, which is the midbrain.
Yeah.
And for those that don't know, the midbrain is an area beneath the cortex. I guess we never really defined cortex. It was kind of the outer layers, or are the outer layers of the, at least mammalian brain or human brain. But the midbrain is super interesting because it controls a lot of unconscious stuff, reflexes, et cetera. And then there's this phenomenon even called blind sight. So could you please tell us about the midbrain, about what it does, and what in the world is blind sight?
Yeah, so there's a lot of pieces there. I think the first thing to say is, if you imagine the nervous system in your mind's eye, you see this big honking brain, and then there's this little wand that dangles down into your vertebral column, the spinal cord, and that's kind of your visual impression. What you have to imagine is starting in the spinal cord and working your way up into this big, magnificent brain. And what you would do as you enter the skull is get into a little place where the spinal cord kind of thickens out. It still has that sort of long, skinny, trunk-like feeling.
Sort of like a paddle or a spoon shape.
Right. It starts to spread out a little bit, and that's because your evolution has packed more interesting goodies in there for processing information and generating movement. So beyond that is this tween brain we were talking about, this linker brain. What diencephalon really means the between brain.
Oh, I thought you said tween.
Well, it is, yes.
No, between. Sorry, it said tween.
Yeah, it's the between. Yeah. It's the between brain is what the name means. It's the linker from the spinal cord and the periphery up to these grand centers of the cortex. But this midbrain you're talking about is the last bit of this enlarged sort of spinal cordy thing in your skull, which is really the brain stem is what we call it. The last bit of that before you get to this relay up to the cortex is the midbrain. And there's a really important visual center there. It's called the superior colliculus. There's a similar center in the brains of other vertebrate animals. A frog, for example, or a lizard would have this. It's called the optic tectum there. But it's a center that in these non-mammalian vertebrates is really the main visual center. They don't really have what we would call a visual cortex, although there's something sort of like that. But this is where most of the action is in terms of interpreting visual input and organizing behavior around that. You can sort of think about this region of the brainstem as a reflex center that can reorient the animal's gaze or body or maybe even attention to particular regions of space out there around the animal. And that could be for all kinds of reasons. I mean, it might be a predator just showed up in one corner of the forest and you pick that up and you're trying to avoid it.
Or just any movement.
Any movement, right? It might be, that suddenly, something splats on the page when you're reading a novel and your eye reflexly looks at it. You don't have to think about that. That's a reflex.
What if you throw me a ball, but I'm not expecting it?
Right.
And I just reach up and try and grab it, catch it or not.
Right.
Is that handled by the midbrain?
Well, that's probably not the midbrain, although, by itself, because it's going to involve all these limb movements, this movement of your arm and body. And
what about ducking if something's suddenly thrown in my head?
Sure, right. Things like that will certainly have a brainstem component, a midbrain component. Something looms and you duck. It may not be the superior colliculus we're talking about now. It might be another part of the visual midbrain. But these are centers that emerged early in the evolution of brains like ours to handle complicated visual events that have significance for the animal in terms of space. Where is it in space? And in fact, this same center actually gets input from all kinds of other sensory systems that take information from the external world, from particular locations, and where you might want to either avoid or approach things according to their significance to you. So you get input from the touch system. You get input from the auditory system. I worked for a while in rattlesnakes. They get input from a part of their warm sensors on their face. They're in these little pits on the face.
You used to work on baby rattlesnakes, right?
Well, they were adults, actually.
Oh, I wasn't trying to diminish the danger.
No,
I thought for some reason they were little ones.
No.
Why in the world would you work on rattlesnakes?
Well, because they have a version of an exteroceptive sensory system. That is, they're looking out into the world using a completely different set of sensors. They're using the same sensors that would feel the warmth on your face if you stood in front of a bonfire, except evolution has given them this very nice specialized system that lets them image where the heat's coming from. You can sort of do that anyway, right? If you walk around the fire, you can feel where the fire is from the heat hitting your face.
Is that the primary way in which they detect prey?
It's one of the major ways, and in fact, they use vision as well. And they bring these two systems together in the same place, in this tectal region, this brainstem, midbrain region.
What's all the tongue jutting about when the snakes?
That I don't know. That may be olfactory. There may be.
They're sniffing the air with their tongue.
There may be.
Earlier in our drive, you told me that flies actually taste things with their feet.
They do, yeah.
That's so weird.
Yeah, they have taste receptors in lots of funny places.
I want to pause here just for 1 second before we get back into the midbrain. I think what's so interesting in all seriousness about taste receptors on feet, heat sensors, tongues jutting out of snakes, and vision and all this integration is that it really speaks to the fact that all these sensory neurons are trying to gather information and stuff it into a system that can make meaningful decisions and actions. And that it really doesn't matter whether or not it's coming from eyes or ears or nose or bottoms of feet, because in the end, it's just electricity flowing in. Sure. And so it sounds like it's placed on each animal. It always feels weird to call a fly an animal, but they are creatures. They are animals. It's placed in different locations on different animals depending on the particular needs of that animal.
Right. But how much more powerful if the nervous systems can also cross-correlate across sensory systems. So if you've got a weak signal from one sensory system, you're not quite sure there's something there, and a weak signal from another sensory system that's telling you the same locations is a little bit interesting. There might be something there. If you've got those two together, you've got corroboration. Your brain now says it's much more likely that that's going to, be something worth paying attention to.
Right. So maybe I'm feeling some heat on one side of my face, and I also smell something baking in the oven.
Right.
So now it's neither is particularly strong, but as you said, there's some corroboration.
Right.
And that corroboration is occurring in the midbrain.
Right. And then if you throw things into conflict, now the brain is confused, and that may be where your motion sickness comes from. So it's great to have, as a brain, it's great to have as many sources of information as you can have. Just like if you're you're a spy or a journalist, you want as much information as you can get about what's out there. But if things conflict, that's problematic, right? Your sources are giving you different information about what's going on. Now you've got a problem on your hands. What do you publish?
The midbrain is so fascinating. I don't want to eject us from the midbrain and go back to the vestibular system, but I do have a question that I forgot to ask about the vestibular system, which is, why is it that for many people, including me, despite my motion sickness in cabs, that there's a sense of pleasure in moving through space and getting tilted relative to the gravitational pull of the earth? For me, growing up, it was skateboarding, but people like to corner in cars, corner on bikes. Maybe for some people it's done running or dance, but what is it about moving through space and getting tilted? A lot of surfers around here, getting tilted, that can tap into some of the pleasure centers. Do we have any idea why that would feel good?
I have no clue.
Is there dopaminergic input to this system?
Well, the dopaminergic system gets a lot of places. It's pretty much, to some extent, everywhere in the cortex, a lot more in the frontal lobe, of course. But, that's just for starters. I mean, there's basically dopaminergic innervation most places in the central nervous system. So there's the potential for dopaminergic involvement, but I really have no clue about the tilting phenomenon. I mean,
people pay money to go on roller coasters.
Right. Well, I think that may be as much about the thrill as anything else.
Sure. And the falling reflex is very robust in all of us.
Right.
When the visual world's going up very fast, it usually means that we're falling.
Right.
But and some people like that, some people don't.
Right. And kids tolerate a lot more, sort of vestibular craziness spinning around until they drop than adults.
I have friends, it always, worries me a little bit that they throw their kids. I'm not recommending anyone do this. When they're little kids, like throwing the kids really far back and forth. Some kids seem to love it.
Yeah. Yeah, our son loved being shaken up and down very, very vigorously. That's the only thing that would calm him down sometimes.
Interesting. Yeah, so I'm guessing we can guess that maybe there's some activation of the reward systems from being moving through space.
Well, if you think about it, how rewarding it is to be able to move through space, and how unhappy people are who are used to that, who suddenly aren't able to do that. There is a sense of agency, right? If you can choose to move through the world and to tilt, that's not only you're moving through the world, but you're doing it with a certain amount of finesse. Maybe that's what it is. You feel like you're the master of your own movement in a way that you wouldn't if you're going straight. I'm just blowing smoke here. Right?
Yeah, well, we can speculate. That's fine. I couldn't help but ask the question. Okay, so if we move ourselves, pun intended, back into the midbrain. The midbrain's combining all these different signals for reflexive action. At what point does this become deliberate action? Because if I look at something I want and I want to pursue it, I'm going to go toward it. And many times that's a deliberate decision.
Right. So this gets very slippery, I think, because what you have to try to imagine is all these different parts of the brain working on the problem of staying alive, and surviving in the world. They're working on the problem simultaneously, and there's not one right answer to how to do that. But one way to think about it is that you have high levels of your nervous system that are very well designed to override an otherwise automatic movement if it's inappropriate. So if you imagine you've been invited to tea with the queen, and she hands you, very fancy Wedgwood, teacup, very thin.
Wedgwood teacup?
Yes, with very hot tea in it, and you're burning your hand. You probably will try to find a way to put that back down on the saucer rather than just dropping it on the floor. because you're with the queen. You're trying to be appropriate to that. So you have ways of reining in automatic behaviors if they're going to be maladaptive. But you also want the reflex to work quickly if it's the only thing that's going to save you. The looming, object coming at your head. You don't have time to think about that. So this is the interplay in these hierarchically organized centers of the nervous system. At the lowest level, you've got the automatic sensors and centers and reflex, arcs that will keep you safe, even if you don't have time to think about it. And then you've got the higher center saying, well, maybe we could do this as well, or maybe we shouldn't do that at all, right? So you have all of these different levels operating simultaneously, and you need bidirectional communication between high-level cognitive centers, decision-making on the one hand, and these low-level, very helpful reflexive centers, but they're a little bit rigid, a little hardwired, so they need some nuance. So both of these things are operating in tandem in real time, all the time in our brains. And sometimes we listen more to one than the other. You've heard people in sports talking about messing up at the plate because they overthought it, thinking too hard about it. That's partly, you've already trained your cerebellum how to hit a fastball right down the middle.
Right. And if you start looking for something new or different, you're going to mess up your reflexive swing.
Right. If you're trying to think about the physics of the ball as it's coming at you, you've already missed, right? Because you're not using all those reps have built a kind of knowledge is what you want to rely on when you don't have enough time to contemplate.
This is important and a great segue for what I'd like to discuss next, which is the basal ganglia. This really interesting area of the brain that's involved in go type commands and behaviors, instructing us to do things, and no go, preventing us from doing things. Because so much of motor learning and skill execution and not saying the wrong thing or sitting still in class or as you used with the tea with the queen example, feeling discomfort involves suppressing behavior. And sometimes it's activating behavior.
Right.
A tremendous amount of online attention is devoted to trying to get people motivated. This isn't the main focus of our podcast. We touch on some of the underlying neural circuits of motivation, dopamine, and so forth. But so much of what people struggle with out there are elements around failure to pay attention, or challenges in paying attention, which is essentially like putting the blinders on, getting a soda straw view of the world and maintaining that for a bout of work or something of that sort, and trying to get into action. So, of course, this is carried out by many neural circuits, not just the basal ganglia. But what are the basal ganglia and what are their primary roles in controlling go-type behavior and no-go type behavior?
Yeah, so the basal ganglia are sitting deep in what you would call the forebrain, so the highest levels of the brain. They are sort of cousins to the cerebral cortex, which we talked about as sort of the highest level of your brain, the thing you're thinking with.
The cerebral cortex being the refined cousins, and then you've got the the brutes.
That's probably totally unfair, but the point.
I like the basal ganglia. I can relate to the brutish parts of the brain. A little bit of hypothalamus, a little bit of basal ganglia. Sure,
we need it all. And this area of the brain has gotten a lot bigger as the cortex has gotten bigger, and it's deeply intertwined with cortical function. The cortex can't really do what it needs to do without the help of the basal ganglia and vice versa. So they're really intertwined. And in a way, you can think about this logically as saying, if you have the ability to withhold behavior or to execute it, how do you decide which to do? Well, the cortex is going to have to do that thinking for you. You have to be looking at all the contingencies of your situation and decide, is this a crazy move or is this a really smart investment right now, or what?
Right. I don't want to go out for a run in the morning, but I'm going to make myself go out for a run. Or I'm having a great time out on a run and I know I need to get back, but I kind of want to go another mile.
Another great example is that, the marshmallow test for the little kids. They can get 2 marshmallows if they hold off, just 30 seconds initially. They can have one right away, but if they can wait 30 seconds, they got 2. So that's the no-go, because their cortex is saying, I would really like to have 2 more than having 1. but they're not going to get the 2 unless they can not reach for the 1. So they've got to hold off the action. And that has to result from a cognitive process. So the cortex is involved in this in a major way.
As I recall in that experiment, the kids used a variety of tools some would distract themselves.
Right.
I particularly related to the kid that would just put himself right next to the marshmallows and then some of the kids covered their eyes. Some of them would count or sing. Yeah, so that's all very cortical, right? Coming up with a novel strategy. Simple example that we're using here, but of course this is at play anytime someone decides they want to go watch a motivational speech or something, just, a Steve Jobs commencement speech, just to get motivated to engage in their day.
Should I take this new job? Mm-hmm. It's got great benefits, but it's in a lousy part of the country.
Why do you think that some people have a harder time running these go, no-go circuits, and other people seem to have very low activation energy, we would say? They can just,
right.
They have a task, they just lean into the task.
Right.
Whereas some people getting into task completion or things of that sort is very challenging for them.
Yeah. I think it's really just it's a special case of a very general phenomenon, which is brains are complicated. And the brains we have are the result of genetics and experience. And my genes are different from your genes, and my experiences are different from your experiences. So the things that will be easy or hard for us won't necessarily be aligned. They might just happen to be just because they are. But the point is that, you're dealt a certain set of cards, you have a certain set of genes, you are handed a brain. You don't choose your brain, it's handed to you. But then there's all this stuff you can do with it. You can learn to have new skills or to act differently or to show more restraint, which is kind of relevant to what we're talking about here. Or maybe show less restraint if your problem is you're so buttoned down you never have any fun in life. You should loosen up a little bit, right?
Thank you. I appreciate the insult.
Yeah.
David's always encouraged me to have a little more fun in life. So basal ganglia they're kind of the disciplinarian, or they're sort of the instructor or conductor of sorts, right? Go, no go, you be quiet, you start now.
I wish I knew more about the basal ganglia than I do. My sense is that this system is key for implementing the plans that get cooked up in the cortex, but they also influence the plans that the cortex is dishing out, because this is a major source of information to the cortex. So it becomes almost impossible to figure out where the computation begins and where it ends and who's doing what, because these things are all interacting in a complex network. And it's all of it. It's the whole network. It's not, one is the leader and the other is the follower.
Right, of course. Yeah, these are all the structures that we're discussing are working in parallel.
Right.
And there's a lot of changing crosstalk. I have this somewhat sick habit, David, of every day I try and do 21 no-goes. So if I want to reach for my phone, I try and not do it just to see if I can prevent myself from engaging in that behavior. If it was reflexive, if it's something I want to do, a deliberate choice, then I certainly allow myself to do it.
Right.
I don't tend to have too much trouble with motivation with go-type functions, mostly because I'm so busy that I wish I had more time for more goes, but so to speak. But do you think these circuits have genuine plasticity in them?
Absolutely. Everybody knows how they've learned over time to wait for the 2 marshmallows, right? You don't have to have instant gratification all the time. You're willing to do a job sometimes that isn't your favorite job because it comes with the territory and you want the salary that comes at the end of the week or the end of the month, right? So we can defer gratification. We can choose not to say the thing that we know is going to inflame our partner and create a meltdown for the next week. We learn this control, but I think these are skills like any other. You can get better at them if you practice them. So I think you're choosing to do that just spontaneously as kind of a, it's a mental practice. It's a discipline. It's a way of building a skill that you want to have.
Yeah, I find it to be something that when I engage in a no-go type situation, then the next time and the next time that I find myself about to move reflexively, there's a little gap in consciousness that I can make a decision whether or not this is really the best use of my time. Because I sometimes wonder whether or not all this business around attention, certainly there's the case of ADHD and clinical diagnosed ADHD, but all these, the issue around focus and attention is really that people just have not really learned how to short circuit a reflex. And so much of what makes us different than rattlesnakes or, well, actually they could be deliberate, but from the other animals, and is our ability to suppress reflex.
Yeah. Well, that's the cortex. I mean, or let's say the forebrain. Cortex and basal ganglia working together, sitting on top of this lizard brain that's giving you all these great adaptive reflexes that help you survive. You just hope you don't get the surprising case where the thing that your reflex is telling you is actually exactly the wrong thing and you make a mistake, right?
And
that's
die.
what, right. So that's what the cortex is for. It's adding nuance and context and experience, past association, and in human beings, obviously learning from others through, communication.
Well, you went right to it, and it was where I was going to go. So let's talk about the cortex. We've worked our way up the so-called neuraxis, as the aficionados will know. So we're in the cortex. This is the seat of our higher consciousness, self-image, planning, and action. But as you mentioned, the cortex isn't just about that. It's got other regions that are involved in other things. So maybe we should, staying with vision, let's talk a little bit about visual cortex. You told me a story, an amazing story about visual cortex, and it was somewhat of a sad story, unfortunately, about someone who had a stroke to visual cortex. Maybe if you would share that story, because I think it illustrates many important principles about what the cortex does.
Right. So, the visual cortex is, you could say, the projection screen, the first place where this information streaming from the retina through this thalamus, connecting linker gets played out for the highest level of your brain to see. I mean, it's a representation. It's a map of things going on in the visual world that's in your brain. And when we describe a scene to a friend, we're using this chunk of our brain to be able to put words, which are coming from a different part of our cortex, to the objects and movements and colors that we see in the world. So that's a key part of your visual experience. When you can describe the things you're seeing, you're looking at your visual cortex. And this is...
Could I just ask a quick question? So right now, because I'm looking at your face,
right,
as we're talking, there are neurons in my brain, more or less in the configuration of your face, that are active as you move about. And what if I were to close my eyes and just imagine, I do this all the time, by the way, David. I close my eyes and I imagine David Burson's face. I don't tend to do that as often. Maybe I should. But you get the point. I'm now using visualization
right.
of what you look like by way of memory.
Right.
If we were to image the neurons in my brain, would the activity of neurons resemble the activity of neurons that's present when I open my eyes and look at your actual face?
This is a deep question. We don't really have a full
Seems like an easy experiment to do.
Yes, except you're talking about looking in detail at the activity of neurons in a human brain, and that's not as easy to do as it would be in in some kind of animal model. But the bottom line is that you have a spatial representation of the visual world laid as a map of the visual world laid out on the surface of your cortex. The thing that's surprising is that it's not one map. It's actually dozens of maps.
What do each of those maps do?
Well, we don't really have a full accounting there either, but it looks a little bit like the diversification of the output neurons of the retina, the ganglion cells we were talking about before. There are different types of ganglion cells that are encoding different kinds of information about the visual world. We talk about the ones that were encoding the brightness, but other ones are encoding motion or color, these kinds of things. The same kinds of specializations in different representations of the visual world in the cortex seem to be true. It's a complex story. We don't have the whole picture yet, but it does look as if some parts of the brain are much more important for things like reaching for things in the space around you, and other parts of the cortex are really important for making associations between particular visual things you're looking at now and their significance. What is that object? What can it do for me? How can I use it?
What about the really specialized areas of cortex, like the areas like neurons that respond to particular faces, or neurons that, I don't know, can help me understand where I am relative to some other specific object?
Right. So, these are properties of neurons that are extracted from, detected by recording the activity of single neurons in some experimental system. What's going on when you actually perceive your grandmother's face is a much more complicated question. It clearly involves hundreds and thousands and probably millions of neurons acting in a cooperative way. So you can pick out any one little element in this very complicated system and see that it's responding differentially to certain kinds of visual patterns, and you think you're seeing a glimpse of some part of the process by which you recognize your grandmother's face. But that's a long way from a complete description, and it certainly isn't going to be at the level of a magic single neuron that has the special stuff to recognize your grandmother. It's going to be in some pattern of activity across many, many cells, resonating in some kind of special way that will represent the internal, memory of your mother. So,
which is really incredible.
Yeah.
I mean, every time we do this deep dive, which we do from time to time, you and I, we kind of like march into the nervous system and explore how different aspects of our life experiences is handled there and how it's organized. After so many decades of doing this, it still boggles my mind that the collection of neurons 1 through 7 active in a particular sequence gives the memory of a particular face, and run backwards 7 through to 1 gives you could be rattlesnake, pit viper, heat sensing organs, as we were talking about earlier. So is it true that there's a lot of multi-purposing of the circuitry? Like, we can't say one area of the brain does A and another area of the brain does B. So, areas can multitask or have multiple jobs. They can moonlight.
Right. But I think in my career, the hard problem has been to square that with the fact that, things are specialized, that there are specific genes expressed in specific neurons that make synaptic connections with only certain other neurons. And that particular synaptic arrangement actually results in the processing of information that's useful to the animal to survive, right? So it's not as if it's either a big undifferentiated network of cells, and looking at any one is never going to tell you anything. That's too extreme on the one hand. Nor is it the case that everything is hardwired and every neuron has one function and this all happens in one place in the brain. It's way more complicated and interactive and interconnected than that.
So we're not hardwired or soft-wired.
It's both.
We're sort of, I don't know what the analogy should be. What substance would work best, David?
No idea there. But the idea is that it's always network activity. There's always many, many neurons involved. And yet there's tremendous specificity in the neurons that might or might not be participating in any distributed function like that, right? So you have to get your mind around the fact that it's both very specific and very non-specific at the same time. It's a little tricky to do, but I think that's kind of where the truth lies.
Yeah, and so this example that you mentioned to me once before about a woman who had a stroke in visual cortex, I think speaks to some of this.
Right.
Could you share with us that story?
Sure. So the point is that you all, those of us who see, have representations of the visual world in our visual cortex. What happens to somebody when they become blind because of problems in the eye, the retina perhaps? You have a big chunk of the cortex, this really valuable real estate for neural processing, that has come to expect input from the visual system, and there isn't any anymore. So you might think about that as fallow land, right? It's used by the nervous system, and that would be a pity. But it turns out that it is in fact used. And the case that you're talking about is of a woman who was blind from very early in her life, and who had risen through the ranks to a very high-level executive secretarial position in a major corporation. And she was extremely good at Braille reading, and she had a Braille typewriter, and that's how everything was done. And apparently she had a stroke and was discovered at work, collapsed, and they brought her to the hospital. And apparently the neurologist who saw her when she finally came to said, I've got good news and bad news. Bad news is you had a stroke. The good news is that it was in an area of your brain you're not even using. It's your visual cortex. And I know you're blind from birth, so there shouldn't be any issue here. The problem was she lost her ability to read Braille. So what appears to have been the case, and this has been confirmed in other ways by imaging experiments in humans, is that in people who are blind from very early in birth, the visual cortex gets repurposed as a center for processing tactile information. And especially if you train to be a good Braille reader, you're actually reallocating somehow that real estate to your fingertips, a part of the cortex that should be listening to the eyes. So that's an extreme level of plasticity. But what it shows is the visual cortex is kind of a general purpose processing machine. It's good at spatial information, and the skin of your fingers is just another spatial sense. And deprived of any other input, the brain seems smart enough, if you want to put it that way, to rewire itself to use that real estate for something useful, in this case, reading Braille.
Incredible. Somewhat tragic, but incredible. Right. At least very in that case, tragic.
Right. Yeah. Very informative.
Very informative. And of course, it can go the other way too.
Right.
Where people can gain function in particular modalities like improved hearing or tactile function in the absence of vision.
Right.
Tell us about connectomes. We hear about genomes, proteomes, microbiomes, ohms, these days. What's a connectome and why is it valuable?
Yeah, so the connectome actually now has two meanings. So I'll only refer to the one that is my passion right now, and that is really trying to understand the structure of nervous tissue at a scale that's very, very fine.
But smaller than a millimeter.
Way smaller than a millimeter, a nanometer or less. That's 1,000 times smaller. Or it's actually, a million times smaller. So really, really tiny. On the scale of individual synapses between individual neurons, or even smaller, like the individual synaptic vesicles containing little packets of neurotransmitter that are going to get released to allow one neuron to communicate to the next. So very, very fine. But the notion here is that you're doing this section after section at very fine scale. So in theory, what you have is a complete description of a chunk of nervous tissue that is so complete that if you took enough time to identify where the boundaries of all the cells are, you could come up with a complete description of the synaptic wiring of that chunk of nervous tissue, because you have a complete description of where all the cells are and where all the synapses between where all the cells are. So now you essentially have a wiring diagram of this complicated piece of tissue. So the omics part is the exhaustiveness of it. Rather than looking at a couple of synapses that are interesting to you from two different cell types, you're looking at all the synapses of all of the cell types, which of course is this massive avalanche of data, right?
So in genetics, you have genetics and then you have genomics, which is the idea of getting the whole genome.
All of it.
And we don't really have an analogous word for genetics, but it would be connectivity and connectomics.
Right. Or
connectomics.
Connectomics, sure.
Connectivity and connectomics.
Right. So it's wanting it all. And of course, it's crazy ambitious, but that's where it gets fun. It's a use of electron microscopy, a very high resolution microscopic imaging system on a new scale with way more payoff in terms of understanding the connectivity of the nervous system. And it's just emerging, but I really think it's going to revolutionize the field, because we're going to be able to query these circuits. How do they actually do it? Look at the hardware in a way that's never been possible before.
The way I describe this to people is if you were to take a chunk of kind of cooked but cold spaghetti,
right,
and slice it up very thin, you're trying to connect up each image of each slice of the edge of the spaghetti as figure out which ropes of spaghetti belong to which.
And have a complete description of where this piece of spaghetti touches that piece of spaghetti, and is there something special there?
Where the meat sauce is and all the other cell types and the pesto, all where it all is around the spaghetti, because those are the other the blood vessels and the glial cells. And so what's it good for? I mean, maps are great.
Right.
I always think of connectomics and genomics and proteomics, et cetera, as necessary but not sufficient.
Right. So, I mean, in many cases, what you do is you go out and probe the function, and you understand how the brain does the function by finding neurons that seem to be firing in association with this function that you're observing. And little by little, you work your way in, and now you want to know what the connectivity is. Maybe the anatomy could help you. But this connectomics approach, or at least the serial electron microscopy reconstruction of neurons approach, really is allowing us to frame questions starting from the anatomy and saying, I see a synaptic circuit here. My prediction would be that these cell types would interact in a particular way. Is that right? And then you can go and probe the physiology, and you might be right or you might be wrong. But more often than not, it looks like the structure is pointing us in the right direction. So in my case, I'm using this to try to understand a circuit that is involved in this image stabilization network we're talking about, keeping things stable on the retina. And this thing will only respond at certain speeds of motion. These cells in the circuit, like slow motion, they won't respond to fast motion. How does that come about? Well, I was able to probe the circuitry. I knew what my cells looked like. I could see which other cells were talking to it. I could categorize all the cells that might be the players here that are involved in this mechanism of tuning the thing for slow speeds. And then we said, it looks like it's that cell type. And we went and looked, and the data bore that up. But the anatomy drove the search for the particular cell type because we could see it connected in the right place to the right cells. That creates the hypothesis that lets you go query the physiology, but it can go the other way as well. So it's always the synergy between these functional and structural approaches that gives you the most lift. But in many cases, the anatomy has been a little bit the weak sister in this, the structure, trying to work out the diagram, because we haven't had the methods. Now the methods exist. And this whole field is expanding very quickly because people want these circuit diagrams for the particular part of the nervous system that they're working on. If you don't know the cell types and the connections, how do you really understand how the machine works?
Mm-hmm. Yeah, what I love about it is we don't know what we don't know.
Right.
And scientists, we don't ask questions, we pose hypotheses. Hypotheses being, of course, some prediction that you wager your time on, basically.
Right.
And it either turns out to be true or not true. But if you don't know that a particular cell type is there, you could never, in any configuration of life or a career or exploration of a nervous system, wager a hypothesis because you didn't know it was there. Right. So this allows you to say, there's this little interesting little connection between this cell that I know is interesting and another cell that's a little mysterious but interesting. I'm going to hypothesize that it's doing blank, blank, and blank and go test that. And in the absence of these connectomes, you would never know that cell was lurking there in the shadows.
Right. Yeah, and if you're just trying to understand how information flows through this biological machine, you want to know where things are. Neurotransmitters are dumped out of the terminals of one cell, and they diffuse across the space between the two cells, which is kind of a liquidy space, and they hit some receptors on the postsynaptic cell, and they have some impact. Sometimes that's not through a regular synapse. Sometimes it's through a neuromodulator, like you often talk about on your podcast, that are sort of oozing dopamine, exactly, oozing into the space between the cells, and it may be acting at some distance far from where it was released, right? But if you don't know where the release is happening and where other things are that might respond to that release, you're groping around in the dark.
Well, I love that you are doing this. And I have to share with the listeners that the first time I ever met David, and every time I've ever met with him in person, at least at his laboratory at Brown, he was in his office, door closed, drawing neurons and their connections. And this is somewhat unusual for somebody who's a endowed full professor, chairman of the department, et cetera, for many years, to be doing the hands-on work. Typically, that's the stuff that's done by technicians or graduate students or postdocs. But I think it's fair to say that you really love looking at nervous systems and drawing the accurate renditions of how those nervous systems are organized and thinking about how they work.
Yeah, it's pure joy for me. I mean, I'm a very visual person. My wife is an artist. We look at a lot of art together. Just the forms of things are gorgeous in their own right. But to know that the form is, in a sense, the function, that the architecture of the connectivity is how the computation happens in the brain at some level, even though we don't fully understand that in most contexts, gives me, great joy, because I'm working on something that's both visually beautiful, but also deeply beautiful in a sort of a higher sort of knowledge context. What is it all about?
I love it. Well, as a final question, I get asked very often about how people should learn about neuroscience, or how they should go about pursuing maybe an education in neuroscience if they're at that stage of their life or that's appropriate for their current trajectory. Do you have any advice to young people, old people, and anything in between about how to learn about the nervous system more, maybe in a more formal way? I mean, obviously we have our podcast. There are other sources of neuroscience information out there, but for the young person who thinks they want to understand the brain, they want to learn about the brain, what should we tell them?
Well, that's a great question. And there's so many sources out there. It's almost a question of, how do you deal with this avalanche of information out there? I mean, I think your podcast is a great way for people to learn more about the nervous system in an accessible way. But there's so much stuff out there. And it's not just that. I mean, the resources are becoming more and more available for average folks to participate in neuroscience research on some level. There's this famous EyeWire project of Sebastian
maybe
Soma.
tell us about EyeWire.
Yeah, so that's connectomics, and that's a situation where a very clever scientist realized that the physical work of doing all this reconstruction of neurons from these electron micrographs, there's a lot of time involved. Many, many person hours have to go into that to come up with the map that you want of where the cells are. And he was very clever about setting up a context in which he could crowdsource this, and people who were interested in getting a little experience looking at nervous tissue and participating in a research project could learn how to do this and do a little bit.
From their living room.
From their living room, their laptop.
We'll put a link to EyeWire. It also is a great bridge between what we were just talking about, connectomics, and actually participating in research.
Right.
And you don't need a graduate mentor or anything like that.
Right. So more of this is coming, and I'm actually interested in building more of this, so that people who are interested, want to participate at some level, don't necessarily have the time or resources to get involved in laboratory research, can get exposed to it and participate and actually contribute. So I think that's one thing. I mean, just asking questions of the people around you who know a little bit more and have them point you in the right direction. Here's a book you might like to read. There's lots of great popular books out there that are accessible that will give you some more sense of the full range of what's out there in the neurosciences and how
we can put some links to a few of those that we like on basic neuroscience.
Right.
Our good friend Dick Maslin, the late Richard, people call him Dick Maslin, had a good book. I forget the title at the moment. It's sitting behind me somewhere over there on the shelf, but about vision and how nervous systems work. A pretty accessible book for the general public.
Right.
Yeah.
Right. So, that and there's so many sources out there. I mean, Wikipedia is a great way. If you got a particular question about visual function, I would say by all means, head to Wikipedia and get the first look and follow the references from there, or go to your library, or there's so many ways to get into it. It's such an exciting field now. There's so many, any particular realm that's special to you, your experience, your strengths, your passions, there's a field of neuroscience devoted to that. If you know somebody who's got a neurological problem or a psychiatric problem, there's a branch of neuroscience that is devoted to trying to understand that and to solve these kinds of problems down the line. So feel the buzz. It's an exciting time to get involved.
Great. Those are great resources that people can access from anywhere, zero cost, as you need an internet connection. But aside from that, we'll put the links to some. And I'm remembering Dick's book is called We Know It When We See It.
Right. One of my heroes.
Yeah, a wonderful colleague who unfortunately we lost a few years ago. But Listen, David, this has been wonderful.
It's been a blast.
We really appreciate you taking the time to do this. As people probably realize by now, you're an incredible wealth of knowledge about the entire nervous system. Today, we just hit this top contour of a number of different areas to give a flavor of the different ways that the nervous system works and is organized and how that's put together, how these areas are talking to one another. What I love about you is that you're such an incredible educator and I've taught so many students over the years, but also for me personally, as friends, but also anytime that I want to touch into the beauty of the nervous system, I rarely lose touch with it, but anytime I want to touch into it and start thinking about new problems and ways that the nervous system is doing things that I hadn't thought about, I call you. So please forgive me for the calls past, present, and future, unless you change your number. And even if you do, I'll be calling.
It's been such a blast, Andy. This has been a great session, and it's always fun talking to you. It always gets my brain racing. So thank you.
Thank you. Thank you for joining me today for my discussion with Dr. David Burson. By now, you should have a much clearer understanding of how the brain is organized and how it works to do all the incredible things that it does. If you're enjoying and or learning from this podcast, please subscribe to our YouTube channel. That's a terrific zero-cost way to support us. In addition, please subscribe to our podcast on Apple and Spotify. And on Apple, you have the opportunity to leave us up to a 5-star review. As well, if you'd like to make suggestions for future podcast episode topics or future podcast episode guests, please put those in the comment section on our YouTube channel. Please also check out our sponsors mentioned at the beginning of each podcast. That's the best way to support us. And we have a Patreon. It's patreon.com/Andrew Huberman. There you can support us at any level that you like. While today's discussion did not focus on supplements, many previous podcast episodes include discussions about supplements. And while supplements aren't necessary for everybody, many people derive benefit from them for things like sleep or focus or anxiety relief and so on. One issue with the supplement industry, however, is that oftentimes the quality will really vary across brands. That's why we partnered with Thorne, Thorne, because Thorne supplements are of the absolute highest standards in terms of the quality of the ingredients they include and the precision of the amounts of the ingredients they include. In other words, what's listed on the bottle is what's actually found in the bottle, which is not true of many supplements out there that have been tested. If you'd like to see the supplements that I take, you can go to thorne.com/U/Huberman, and there you can see the supplements that I take, and you can get 20% off any of those supplements. And if you navigate deeper into the Thorne site through that portal, thorne.com/U/Huberman, you can also get 20% off any of the other supplements that Thorne happens to make. If you're not already following Huberman Lab on Instagram and Twitter, feel free to do so. Both places I regularly post short video posts or text posts that give tools related to health and neuroscience and so forth. And most of the time, that information is non-overlapping with the information on the podcast. Again, it's just Huberman Lab on Instagram and Twitter. And last but not